Yes, good evening to all the participants. So, welcome to the day one session on Introduction to Biological Databases in the online internship and certificate course on artificial intelligence and machine learning approaches of drug and vaccine design. Myself, I am Professor PK Praveen Kumar from Department of Biotechnology, Sri Venkateshwara College of Engineering, and I'm also the Managing Director and Chief Executive Officer of Phyto Nanos Drug Technologies Private Limited. Now, let me start ahead with the today's session. So, actually, how I'm going to take the session for each and every day, the first 5 to 10 minutes, I recap the whole session contents, and then I'll be just exploring with the theoretical concepts. Later, I'll be coming across with the online tools and databases demonstration. Okay, so this is how I'm going to take you the sessions, and I'll be sharing you the recorded videos on the same day. Okay, on tonight you'll be receiving under unfortunate conditions, you'll be receiving it on the next day morning. Okay, so this is how you'll be getting this. And related to the participants, some instructions like actually I have sent you the invitation in Google Calendar. So whatever registered email IDs you have registered, in the registered email IDs, I have sent you the Google invitation. How you receive? No, you will be receiving the invitation like this. So invitation, internship, and certificate course. You have to click here, and here you have joined with Google Meet. But once you have to click yes. If you click yes, okay, the thing is changed now. So if it is changed for each and every time, there is no need to admit personally. Okay. Like if I want to teach something and if you're joining it between, it disturbs the other participants also. So I suggest you to click yes. Now after clicking yes, you can say this. So for each and every day at 6:44 p.m., you will be getting a reminder to join with Google Meet. So you can join directly like that. However, in the WhatsApp group, whatever you have joined, okay, I have also sent the Meet link. So if you want to join with the Meet link, after clicking only once, this you have to click only once for all the 15 days session. Only 14 days you have the session, and 15th day is just for assessment, MCQ. Okay. So this is how we have this kind of an online internship and certificate course. And related to this, so let us go ahead with the today's session contents. So meanwhile, I can also interact with the products of the company and services of this company. So first we go ahead with the chapter one, biological databases. So to not to interfere, with the audio and video quality, I am stopping my video at present. So now I am going to present the on biological databases. So first I'll be explaining you about the phyto nanostruct technologies. So what are the products and services, approved courses we are conducting, which is approved by MSME, and we'll be discussing on the today's session contents like biological databases, repurposing databases, drug discovery and development, target discovery, target identification, target deconvolution, target verification, target validation, databases in network pharmacology, and target identification databases. Actually, in this internship and certificate course, nearly 28 different institutes are participating in this course. So, institutes, reputed institutes like IIT, VIT, SRM. So, whatever in and around India, actually, if you see more than 13 to 14 states or participating in this internship and certificate course. Okay, so now let me go ahead with this. About Phyto Nanostruct Technology Private Limited. It is an AICT and Ministry of Education Institution Innovation Council. Okay, both are Government of India bodies. It's sponsored by them. And of course, this is recognized by DPIIT, Startup India, and MSME. Okay, this is a healthcare startup established on December 2024, incubated in SVC, Sri Venkateshwara College of Engineering, Sri Perambudur. So we dedicate to support the patients suffering from almost all the diseases, especially cancers, metabolic, stress-related disorders. So these are the types of diseases my PhD scholars are working at present. And previously, I worked on autoimmune diseases and also rare diseases. So we try to develop the precision medicine from the plant source. Okay, about the various products of from the plants we'll be discussing in the future presentations and as well as in the future slides. We actually utilize the novel drug delivery technology. Instead of relying on single technology, we focus on multiple drug delivery technology. And of course, our objective is to bring innovative, sustainable solutions. And this is a sponsored startup, as I earlier mentioned, by AST and MIC. Our company vision is to revolutionize healthcare through plant-based nutraceuticals, phytochemicals, by pioneering nanodrug solutions through advanced nanotechnologies, empowering precision medicine. So, other than that, we are also focusing on mitochondrial drug delivery technologies and related to the manufacturing division in phytonanos. So, we are synthesizing and offering nanodrugs of various levels of purity above 50 percentage, above 80 percentage, and above 95 percentage HPLC pure. If the prices are not affordable for procurement to undergo research, okay, whatever even we are selling these phytochemical compounds, secondary metabolites, phytochemical compounds, okay, these are in research chemical grade. It means that we can use these kind of a products up to checking in vitro and in vivo experiments. That is in vitro checking in cell lines, in vivo checking with small animals and rodents like rats, mice, hamsters, etc. So whatever research chemical grade nano drugs, okay, so we synthesize is around 26 products, okay, 26 secondary metabolites from 7 different plants, okay. So I think we have 7 different plants from 26 metabolites. And other than offering this kind of secondary metabolites, also secondary metabolite conjugated nanodrugs, we are also offering root and root powders. Okay, so in SVC soil campus, we are growing the roots of these herbal plants, and we are also selling these kind of plant-based products. Training division in phytonanos. So we are training in almost the related kind of internship and certificate courses with respect to our company, whatever we are handling in the manufacturing division. So we'll be having much of a hands-on session. That is, I'll be taking you on to the tools and databases. So whatever the videos I'll be sending you, the recorded videos that you can watch, and you can multiple play and pause, and you can learn something, and you can utilize in your research project. So till now, whatever training skills I have provided, the research scholars are utilizing well, and they are completing the PhD. So even this is sufficient enough to complete even the PhD. So only thing is that how you try to uh, produce in the form of an outcome with the research skills, whatever I did, that matters the most. Okay, and if you see this training course, okay, so majorly I am focusing. I've focused on computer drug and vaccine design, artificial intelligence, machine learning, and drug and vaccine design. Already these batches are completed, okay, in the June, July month. And also I have trained on bioinformatics. Okay, so these are the courses I have trained. More than I've trained around 250 participants. But before beginning this kind of a startup, I had an earlier experience in training these kind of a certificate courses to a company called IOI, Institution of Innovations, Thiruvannamalai. So in that, I have trained more than 10 certificate internship and certificate courses. So I have trained more than 2,500 participants in these courses. Okay, so I was training these kind of a courses for the past 5 years. So many students, okay, including I mean the many students and faculties from arts and science, pharmacy, medical, MBBS, MD students, and even the engineering colleges, IIT professors, directors are also joining in our course, and they are acquiring this kind of continuous learning process. So, of course, we are also offering the food donation from our so this is a kind of an thing we are making in addition. And I'm also a Global Virtual Healthcare Summit 2021 speaker. So after this session, I'll be circulating these kind of an YouTube videos. So you can also watch the videos, like whatever video I just presented, this is actually a student's competition, okay, related to students competition, where my team of students have won around 3.6 lakhs in the phase one round of drug discovery hackathon, which was conducted in 2020. So related to that, I have just given a kind of a talk, and these two persons, actually he is actually director of more than 1,000 crore company, I believe. Okay, it's a kind of a software company, and I just as I am the one day academician, I believe I just presented along with this kind of a reputed industrialist, and also one more kind of guest lecture I provided for Dassault Systems Altem Technologies. So I also going to circulate this kind of an YouTube video. Whatever YouTube videos I'm going to circulate to you, these are more related to our kind of a internship and certificate course. And usually I deliver guest lectures. So each and every year I deliver around 80 to 85 external guest lectures to various institutions. And related to the sponsored projects, so this is the recent project I completed from IIT Madras. So this is actually a project funded by ANRF, a TARE scheme. So this is a kind of a postdoctoral associateship. So I just worked on in silico, in vitro, and in vivo studies on the effect of plant compounds like methanoid A and vitaferin A. So these plant compounds are steroidal lactone compounds. Okay, so these are steroidal lactone compounds from a plant called ashwagandha, Withania somnifera. And I focused on a mitochondrial kind of a targeting, and I am specializing in this kind of a cancer, hepatocellular carcinoma. And I just completed this postdoctoral associateship under the guidance of Professor Michael Gomiha, who is India's number one bioinformatician in India. And related to the sponsored projects, this is a sponsored project from my first PhD scholar in 2021 in Innovation Protein Challenge. So she has won out of how many 750 plus participants. Nearly 20 participants have received the prizes. So all the 20 participants have received 1.07 lakhs each, and she is the topper out of all the 20, the remaining 19 participants in this innovation protein challenge. Of course, she has given immense pride to us, and these students have secured this kind of an grant, I believe, in 2023 from the Tamil Nadu State Council for as a related kind of a title, whatever I worked in ANRF Tare project in IIT Madras. And this is the project actually drug discovery hackathon. The students have worked on identifying covalent spike protein inhibitors to control the infection of SARS-CoV-2 from small molecule databases. Okay, so we identified a solution for with the plant-based molecules to treat SARS-CoV-2 infection. Okay, so this is what a simple problem statement we have chosen, and of course these students have secured these kind of a grant. And this is one more sponsored. Whatever innovations I am showing, all are related to our kind of a title of the internship and certificate course, and this happened in this decade. It means that 2020 to 2026. Okay, and but actually this startup is a product of AACT. Okay, so the AACT and Ministry of Education Innovation Cell as sponsored. I already mentioned you. We have received around 5 lakhs after winning the phase 4 event pitch deck. In a scheme called Yukti Innovation Challenge 2.0. Okay, so the under the Yukti Innovation Challenge, the Indian government is sponsoring. That is ASD, Government of India is sponsoring around 1 to 10 lakhs. Okay, so based on the budget, they will be fixing this kind of a financial grant. But part of the research innovation I have already completed in IIT Madras, so they have reduced the financial grant of 50%, and they have provided 5 lakhs, not only to work on the innovation, also to establish a kind of a startup and continuing these services. Only 135 innovations secured such a kind of a grants out of 1 lakh 5,000 submissions. And even in the phase 4 round, from phase 3 to phase 4, all the Smart India Hackathon winners, around 200 Smart India Hackathon winners, also merged along with us to give a competition. Nearly 256 winners are selected in phase 3. So 200 and 256, totally 456 participants have given a competition, and out of 456, only 135 innovations have secured the grants. Okay, and fortunately, we are one of the startup, and I am the only professor in Tamil Nadu who received this kind of a grant to establish a kind of a startup. And feedback from participants. I have conducted these kind of a courses earlier. So whatever we are committing, okay, so whatever the participants are participating, we are getting a good kind of a feedback around 100 percentage, and in both the kind of a certificate courses, and the average rating, 5-star rating, we are getting not less than 4.5. Okay, so in AIMLDVD, we got a huge kind of a response, overwhelming response around 4.62. Let me go ahead. Okay, so also I hope I can able to convince you in this kind of whatever you are anticipating from me. Now let me go ahead with this kind of today's session, biological databases. In biological databases, we see the basic databases which is useful from the first session to the last session. So the most useful database, is the most publicly available in the internet. So everyone know. So even the father of bioinformatics, sorry, the father of bioinformatics come from only NCB. So whatever I am telling like a father, she is not a father, but she is a mother. So who is that? Margaret Dayoff. So Margaret Dayoff, who is one of the director of NCBI, okay, and we also consider as one of the pioneer of bioinformatics in the world. Okay, she has established this kind of database, that is National Center for Biotechnology Information. This is hosted by National Institute of Health. And much of the participants or having a PhD background, and even so fewer faculties. Okay, so and also for few or the BTech and few or MSc students, I believe. So this is what I found from your profile, the participants' responses. So if you don't understand, okay, you can ask me at any time. Okay, so you can either unmute and ask me, or you can type the message in the chat box. So in between, if I'm getting out of from the screen, I look through your messages and I can discuss with you. Okay, so don't keep any kind of hesitation. So you have a lot of doubts. Only you have enrolled in this kind of a course, even though you're at a PhD. So whatever you want to discuss with me, you are welcome to discuss at any time. Okay, so I just started with the NCBA PubChem. So PubChem is a chemical compound database from NCBA, National Center for Biotechnology Information. So this is a largest database, not only for the chemical compounds like plant compounds, can also find for drugs, synthetic compounds, etc. Majorly, we use it for the drugs, that is compounds, and we use for substances, and even we can search other than giving a keyword like a compound or drug, we can also give a keyword like a receptor. Here the EGFR stands for epidermal growth factor receptor. This is, of course, a important kinase inhibitor for a cancer. So even we can type the repeated target receptors as the inputs. So whatever inputs we feed, or not only we can feed the target receptor, We can also feed the smiles format. See the smiles format, and a PubChem has a unique kind of an ID called PubChem ID. So even it can feed the PubChem ID as one of the key identifier to retrieve the kind of a data. So the information related to the data are present below. Okay, and this is the URL. Now let me take you to the drug bank. So the drug bank is another kind of a chemical compound database. This is majorly you can find the drug molecules in this drug bank. Okay, whereas in this NCBI PubChem, you find the chemical compounds as well as you can find the drugs. So whatever drugs available in the market, you can able to search either through typing the target, or you can type the pathway, or you can type the drug names. Okay, so these are the keywords we can mention, and this is the URL for the drug bank. And the next is the CHEMDL. So in CHEMDL, we can able to find the compounds information, and the URL is mentioned here. Okay, the compounds drugs we can search in CHEMDL. And in NCP, we have multiple databases. The well-known is a GenBank. The GenBank is majorly for nucleotide. So whatever is a nucleotide, okay, database, we call it as a GenBank. We can find DNA as well as RNA. So both the molecules are present. Okay, so in DNA, we can find linear DNA. We can also find circular DNA. So these are the molecules we can able to find in this nucleotide database. That's what we call it as GenBank. Similarly, if you want to search proteins, instead of nucleotide, we have to click protein and we have to type the keyword like a protein name. Whatever protein name you can give, even you can give that as a keyword for searching the nucleotide. If you want to fetch a DNA or RNA, you can provide that. Or else we can provide the key identifiers like accession number, okay, and gen info ID. So whatever accession is gen bank accession number, and gen info ID is another ID. Okay, so these are the identifiers along with the protein name. And of course, in the manage filters, we can also select the species name. If you want to select specifically the species along with the protein, so we can able to select using by manage filters. And whatever protein we can able to fetch from the NCBI, that we call it as GenPept. GenPept is exclusively for proteins. Okay, but for GenPept and GenBank, commonly we have GenBank accession number. So if even though GenPept a separate name is there for proteins, okay, the kind of an identifier what we use for fetching the information is GenBank accession number and GenInfo ID in both the cases, in both nucleotides as well as proteins. Okay, now let me take you to the next database, a protein database. This is a fully annotated protein database. Fully annotated. What do you mean by fully annotation? It means that if any of the researcher or doing a research from the beginning, if they don't know anything about a specific target, and if they want to explore and they want to do up to a PhD research or PDF research, the most important database we have to search is the UniProt. The UniProt is hosted by UK, like how the NCB is taken care by NIH USA, and UniProt is maintained by UK government. Okay, and in UniProt we have several databases like UniProt Knowledge Base and UniPark. Okay, that is UniProtein Archive and UniReference, and we also have proteomes. Okay, so when I take you to the website, we can go through this kind of a databases in the UniProt. Okay, the detailed annotation you can able to see even in the left side tab panes. Now let me take you to the structural database. Till now, whatever we have discussed is something related to the sequences. Of course, structures are also there in the hyperlinks, but the major structural database for a protein target is PDB. So here you find a maximum number of populated 3D structures of protein from which is established from gold standard techniques. The gold standard techniques like X-ray crystallography, NMR. Okay, so these are the popular techniques utilized. The scientists have utilized to deposit around 2.2 lakh structures. Okay, so whatever 2.2 lakh structures are established, we can also see 10.68 lakh structures below. So this is related to AlphaFold. Okay, so using AI, uh, this kind of a database is established. So around we have 13 lakh structures at present. At present, we have 13 lakh structures present in PDB database. Okay, so this is a database for protein structures. It means the 3D structure of proteins can be downloaded from this database. Okay. AlphaFold. We also have an exclusive URL for AlphaFold. Like however you find the AlphaFold, can you able to see this? AlphaFold DB is what deposited in this computer structure models, okay, which is also available in RCSP. Other than rcsp.org/pdb, we can also see this alphafold.eba.ac. So this is taken care by UK government. Okay, even UK has this kind of an AlphaFold database where you can able to search the protein structures through protein ID, that is PDB ID. The PDB ID is generally the four-digit alphanumerical letters. A PDB ID is generally four digit alphanumerical letters. The next structural database what we search is the NCBI molecular modeling database. Even in NCBI, we have a structured database. We call that as molecular modeling database. So in NCBI, if you select the structure and if you type any PDB ID, obviously it directly take you to this kind of a database where you can download the 3D structure. To download the 3D structure, we have to click here and for finding this information, you can find that. We also have an online visualizer, structure visualizer in NCBI itself, okay, to visualize the structure. And this is another kind of a structure database for a target called EBI, European Bioinformatics Institute, PDBE. Okay, so this is another protein structure database. Okay, here also we can able to download the 3D structure. How to explore it? Just identify search through a four-letter PDB ID. Just type and search, you'll be getting this information. Okay, so to download, you can use the download options. The next database what we'll be discussing. So till now, we have completed initially sequence databases, structure databases. Now we'll be discussing on binding sites databases. If you have ligands and if you have receptors, one should know the binding sites, isn't it? So for this, we can go to a kind of a first database is bindingdb.org. So here you have to type the keywords. Okay, here if you see this is whatever I searched is heat shock protein 75, which we also call as TRAP1, tumor necrosis receptor associated protein 1. This is, of course, a target protein I'm working in my lab, okay, along with the heat shock protein 90. So, heat shock proteins are the major targets of interest in my lab. So, if you want to search specific inhibitors of any targets, so these I am taking as a cancer targets, and whatever you want to search, okay, we can able to search the binding information here using this binding DB. And this is a popular database for protein-protein interactions. So if you want to search on protein-protein interaction networks, this is the largest database string. Okay. In the string, you can find around more than 20 billion interactions data or deposited. How these interactions data are deposited? One is through wet lab techniques. Okay, like however we've seen the PDB structures of proteins or deposited through X-ray and NMR wet lab techniques. Similarly, even this kind of interactions. So it's a great, isn't it? A great advancement. So if you see the number of proteins, 67.6 million proteins, and these are obtained from around 14,094 organisms. Okay, so simply you have to click search and you have to type a keyword of a protein target. So whatever the hub protein target you are searching, the connecting proteins for that hub protein you will be finding from this string database. Of course, this is most important in case of systems biology approach. If you want to go for a precision based medicine, of course, one should have a knowledge of systems biology, that is, learning about genomics, how to integrate genomics, proteomics, interactomics, etc. Of course, this can give you a good knowledge of proteomics. Okay, so one should have this kind of a knowledge to come up with a kind of a precision based medicine. Data sources. So in string database, I have searched a target called HSP90 alpha. Whatever I searched, okay, so the related 10 important interacting proteins of this hub protein. So whatever I searched is now considered as a hub target protein, and whatever interacting proteins there, we can see the top 10 interacting proteins. And of course, these top 10 interacting proteins are selected based on positive regulation as well as negative regulation. Some proteins can uh, give a kind of a potential effect or can give a kind of a negative effect. Okay, whether is it a positively regulating the expression or negatively regulating the expression. So this kind of interactions also we can able to see in this kind of a string database. Okay, so here you can able to see this 10 proteins, okay, both positive and negative regulation information you can able to see, and similarly can increase the number of interactions. Okay, some proteins have around 1,700, 2,500 interactions. In that case, if you want to take all the interactions and if you want to screen based on a particular workflow, okay, or that is based on a particular regulation. So all these things we can able to make use of the string database. Okay, so not only we can able to see the data in the online database string, also if you want to download and open it in a visualizer, that is possible. We have an option called send network to cytoscape. So cytoscape is a free network visualizer. So in this network visualizer, one can able to visualize the protein-protein interactions, I mean DNA-protein interactions. So any kind of interactions we can able to see, the biomolecular interaction we can able to see using the cytoscape. In this case, we can visualize the protein-protein interactions and we can directly upload, opening it using the cytoscape using that option. So, like string database, can also visualize the PPI, that is protein-protein interactions, through a database called FunCoop. Okay, through a database called FunCoop. It looks very user-friendly kind of a thing. Okay, it's a looks very interesting if you go on search for a particular target. So only they are asking like, I mean, they just named as a FunCoop, and here simply you have to search through a particular target. Okay, whatever particular target we search, we can able to see the interacting proteins. We can filter it, we can based on, and we can go for an enrichment. Okay, all these things are possible. The enrichment here, I mean the enrichment based on pathways, enrichment based on expression, everything. Okay, so we can able to make use of this kind of a data. This data can be downloadable. So using this, we can able to understand about the protein-protein interactions and PPI interactions. For this, the HTTP, this is the URL eba.ac.intact. So in this intact, we can able to see this protein-protein interactions. Okay, so this is another database. The next is database of interacting proteins. Okay, this is another database for this dip.doe-mba.ucla.edu. This is the URL. So here you have to search for a particular target, and for a particular hub protein target, we can able to see the interacting proteins information, the protein name, description, etc. All these things are found. The next database, what we are searching here is the BioGrid. Okay, the BioGrid is a database of protein, genetic, and chemical interactions. Okay, like a drug protein interactions and something related to genetics. So this information is also available in a database called thebiogrid.org. Okay, so many institutes are helping as a partners to increase the data in this database. Okay, of course, there's also an useful database, and PPI interactions can also be found in BioGrid. If you search a particular target, like whatever I initially mentioned, heat shock proteins and if I also called as TRAPN, so type any kind of a keyword and it can search a similar kind of an information. Around 480 interacting proteins are available for this trap one. And what are the interacting proteins? This is protein 1, protein 2. Like this, we can see this information of the proteins and we can also see their interaction, expression, etc. Okay. The next database, till now you see, so I just started with sequence database, structure database, then gone with the PPA database. Now I am coming to the, after the binding site database, I've gone to the PPA interaction database. Now I'm coming to the secondary databases. You know, in bioinformatics, the biological databases are classified as primary, okay, primary, secondary, also called as tertiary. I mean, secondary or derived, secondary or derived database. And the third is tertiary or composite, tertiary or composite. Okay, so these are the major broad classifications of databases. The whatever I mentioned you, like NCBI, GenPept, GenBank, okay, these comes under primary databases. Okay, of course, the structures, etc. These are something primary information, isn't it? The structured databases. But if you see this secondary or derived, you can able to see a part of the information of a primary, a part of primary data. That's what we get in secondary database. So what do we call this as derived database? Then what is the part? The part is nothing but a domain, or it can be a motif. What is domain actually? A part of a protein which gives a structure or function of a protein, isn't it? That's what we call it as a functional domain or a structural domains. And similarly, the motifs we call it as functional motif, structural motif. Also, we have sequence motif. Okay, but whatever a part of a thing we consider, okay, and which is most important, so that's we take it separately. Okay, so if and if you have a primary databases, we can able to develop the secondary or derived databases. And the third, what we discuss is the tertiary or composite. The tertiary or composite is something like a hybrid between the primary and secondary. The examples are OWL, ProSite. These are some of the databases comes under tertiary composite. Now let me go ahead with this secondary database. So now we are discussing on the first database called SCOP. SCOP stands for Structural Classification of Proteins. Structural Classification of Proteins. So here you can able to see the proteins are classified under 7 different classes. Okay, the proteins are classified under 7 different classes, and proteins can be searched. That part of that protein domains can be searched through scope ID, SCOP ID, PDB ID, or also Uniprot ID. So these are the key identifiers you have to provide here to search for this scope results. Now let me take you to the secondary database called CATH. CATH stands for Class Architecture Topology Homology. This is also a secondary or derived database where you can able to see 151 million protein domains or classified under 6,573 superfamilies. Here, whatever proteins we can able to see, the protein domains we can see, these are classified under four domains. Okay, like however, I mean four classes. Four classes. Okay, whatever four classes you can see, like however you see, we can 1 minute. I can take an eraser. Yes, now I can take the pen. So we are going to classify the proteins based on the four classes. So one such class is the superfamily. Okay, like this we have family, etc. So these proteins or domains we can able to see from here by typing the keywords like PDB code, gene ontology term, and even the keyword like a protein names. So this is how you can use the CATH. So more about the types of data you can able to see here. Okay, so these are the data I can able to see from CATH. The next thing what we see is the pathway database. The largest pathway database is KE is available from KEGG. KEGG stands for Kyoto Encyclopedia of Genes and Genomes. So this database is from Japan. Okay. So we have multiple databases for multiple databases in KEGG, and one such the popular database what we can see is the pathway database. Okay, and if you search on a particular keyword, here I typed a keyword like a hepatocellular carcinoma, okay, a type of a disease. We can able to see a network of proteins, like what are the proteins are in the pathway and how these protein targets can contribute this to this disease. So for this disease, they have so many. The patients have so many kind of symptoms, like immobilization and propagation, proliferation, differentiation, survival of the cancer cells. So whatever kind of mechanisms. Okay, so here you can also see in the hepatocellular carcinoma, even the pathway mechanisms are mentioned. P53 signaling pathway, tumor growth factor beta signaling pathway, WNT signaling pathway. For each and every pathway, what are the proteins connected to this, and what kind of biological mechanism is happening? So that's information you can able to see in this pathway database. And this was something about repurposing databases. Um, Actually, my voice is audible or not? Can someone interact with me? Because if I found any internet disconnection problems, I don't know. I'll be taking a. And,
Yes, sir, it
uh,
is audible. So
Yes,
from your
sir,
side.
you're audible.
yeah, thank you, thank you. Always keep in touch like this because we don't know what is going to happen in future. But till now, there are no problems. Okay, repurposing databases. So in from next class onwards, I'll be interacting with you in between. I'll be asking you few questions, and you can also answer for that, so that we can make more active sessions,
Sir,
not dull
um,
sessions. Yeah, yeah,
sir, I have a simple query. I'm not sure whether that question has been answered or not. Sir, these
yeah.
slides
So
are very beautiful and
yeah.
very useful also. So will we be also getting the slides to refer on the later on stages?
No, 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 no. It's very clearly mentioned only the videos or session. Video sessions, recorded video sessions. I'll be sending you. You can prepare your own slides. Okay. So whatever
Okay,
I
sir. Okay,
prepared
sir.
is from my yeah. So just follow my recorded because till now I'm just explaining only theoretical, practically like how I go to the tools, how to go explore to the databases. So all these things also will be going in between. So or you can practice, that will give a kind of a best experience. Just by teaching theoretical and just giving the slide information won't give you the practical skills, I believe. Okay, now let me, but
Okay, thank you. Thank you.
whatever hyperlinks I provided, so I'll be pasting in the WhatsApp. So enroll in the WhatsApp group. So whatever links, whatever demo I'm providing, okay, so that I'll be pasting. So that links you can follow, and also follow my videos, so that you can create your own kind of a slides. Okay, similar kind of a slides. Now let me take you to the chemical structures. So just a recap actually. PubChem, this is the URL. Drug Bank, this is the URL. ChemSpider, CS. So these are whatever four things I've shown is related to the chemical structures. The chemical structures here, I mean 1D, 2D, 3D. So we can download all this 1D to 3D information from this chemical structure databases. Similarly, if you want to go with the drug target information, okay, so I told you drug bank, we can download the chemical structures, isn't it? Other than the downloading the chemical structures, if you want to find the drug binding targets, that information is also available in the drug bank. There are plenty of informations available. Of course, this is also a fully annotated database. This is a fully annotated annotation databases, like how I explained you about Uniprot. Okay, and similarly, can able to see the next database is a therapeutic target database. This is the URL for that. Pharmacogenetics knowledge base, drug map central. So whatever the databases, the four databases I have shown, is related to the drug target information. So related to drug design, one should have knowledge on these kind of a databases also. The next thing is a protein interaction information. So just merely doing talking is not sufficient to publish high quality research papers. So our objective is even to learn few more skills related to the computer aided uh, drug design. Okay, and especially if you incorporate the contemporary researchers of system biology approach, that will be better. So for the system biology approach, nowadays the scientists are focusing on the PPI kind of a databases to increase the population, and even to use the kind of a PPI network information to publish in the quality papers. So one such is the HBRD biograde, the biograde biological general repository for interaction. Okay, this is the expansion, and DIP already mentioned, database of interacting proteins. And we also seen few more things, isn't it? String DB, okay, string DB, and then Stitch. So these are some of the databases for the protein interactions. And for pathway, we can go with the NCA pathway, okay, interaction database, NCA PID, okay, and then KEGG, Kyoto Encyclopedia of Genes and Genomes. This is available in genome.jp. KEGG is one of the database in genome.jp. And pathway commons, okay, this also you can able to find related to disease pathways, metabolic pathways, and the uh, and different kind of other pathways. Clinical trial information and adverse effects, clinicaltrial.gov. Okay, so if you want to work on clinical trials, this is the most important website is clinicaltrial.gov. Of course, we also have this clinical trials present in NCBA itself. This is not actually, this is an exclusive database, but clinicaltrial.gov is something different. Okay, it's also we have clinical trial databases in NCBA, adverse reaction database. Of course, this is also available in NCBA. So linked with NCBI, linked with NCBI. Okay, FDA.gov drugs, CIDR. For CIDR, we have side effects. For omics data, we can go with the GUO, sequence read archive, cancer cell line. Okay, also we can able to see, till now we have just whatever we have discussed, we have seen a kind of a recap. Now we'll be seeing something related to a particular cancer. Like if I want to go with a working on a cancer, how to search. We have an exclusive databases like mycancergenome.org. If only if you're working on the cancer, you may need these kind of a databases. Here, this database can give you a good information on the data sets available. From the data sets, you can able to know the patients are suffering from what kind of a gene, what is a significant gene for a particular cancer. Okay, so any cancer information you can able to see in this mycancergenome.org. Now let me go to this DBMHCC. This is a comprehensive HCC biomarker database. Of course, for HCC alone, you can able to focus here on oncodb.hcc. So like this, we have plenty of thousands of databases available. The most important alone we have to choose and we have to work. Okay, because getting sufficient knowledge on this course can also give you a kind of a pathway like how to proceed. Now, if you see an Indian scientist called Raghava from IIT Delhi, okay, in the Indraprastha University, I believe, from Delhi. So he has brought so many kind of a databases for the drug and vaccine design. He also got one kind of a database called Cancer Liver. So here we can able to see an exclusive kind of a data, the clinical kind of a data information, how these data can be used further for discovery of biomarkers and drug design, etc. The next we see is the C Bio portal. In C Bio portal, this is of course a largest database related to cancer. So if anyone want to work with the cancer, I suggest you to go with this kind of a C Bio portal because you'll be getting the molecular profile. So you'll be getting different molecular profile and also how many patients are affected because of hepatocellular carcinoma and on what gene is responsible. Because if you rely on a particular most frequently occurring gene, it will be helpful for you to cure many cases. Okay, so of course, for this you can use that My Cancer Genome C Bio portal. So many statistical data information is available in C Bio portal Cancer Genome. And the next is online Mendelian inheritance in man. See, after completing the table of data, I am now explaining about the disease databases. Okay, I just explored you on the disease pathway database. Now I'm coming to the cancer database. And after the cancer, I'm coming across the genes and genetic disorder database. OMIM. It stands for Online Mendelian Inheritance in Man. Okay, so Online Mendelian Inheritance in Man. So this here you can able to find the what are the genes and what are the genetic disorder diseases information. Okay, this information you can able to search here, and of course for this you have OMIM ID. And we'll next we'll be seeing is the Genomic Data Commons data portal. So of course this is also a cancer database. This is maintained by National Institute of Health. So simply we abbreviate as GDC. GDC is nothing but Genomic Data Commons. So you can able to see the types of cancers, okay, and how many cases. You can see the data is gradually upgraded. Okay, so it's going to. So at present you can see this much number of cases, primary sites, projects, etc. And GEO, gene expression omnibus. Okay, this is related to the gene expression. Generally, how we get this gene expression data, we can get from an microarrays. So before RT-PCR, we are relying on microarrays. The microarrays can provide you the plenty of gene expression data. So generally, we take from mRNA, and from mRNA, we go with reverse transcription to convert to cDNA. So the cDNA expression is what we're measuring, isn't it? In various types of diseases. So in this case, I just search for hepatocellular carcinoma. You can search on any kind of a disease here in this GEO profiles, of course, available in NCBI. So in this, you can able to find this data sets, And using these data sets, whatever you can download, you can use the tools and you can find the expression patterns and you can explore the statistics on that. Now, let me go with the next database called gene cards. I suggest uh, you to go with the gene cards if you're working on any diseases. I presently search for hepatocellular carcinoma. The objective why I'm asking you to use the gene cards, if you're working on diseases, because this gives an extensive information of human genes. For hepatocellular carcinoma, I got around 8,100 genes are responsible for causing this hepatocellular carcinoma. Then what is the major gene and what is the score? See, TP53, tumor protein 53. This is the most occurring gene for hepatocellular carcinoma. So only we can find the score. Of course, even in this database, one can get fully annotation information of that particular genes. So if you just explore, expand it, okay, you can get more information of that particular gene. So you can search the disease, you can search about a particular gene, and you can get much more information related to that protein interaction, etc. Okay. Of course, I suggest you to go with the gene cards if you work on any diseases. The Cancer Proteome Atlas, okay, the TCPA portal. So if you're working on proteomes, okay, so this is actually proposed by MD Anderson Cell Lines Institute. So in the project, you can able to see this PPA interactions. Each and every dot is a protein. So proteins are interacted in this or present in the form of nodes in the network. So for each and every dot is a protein and how it's connected to the network. This is how we can able to see a kind of a network in TCPA portal, the Cancer Proteomic Atlas portal. The next is the open targets. So here we can able to find the target information. Like if you focus on a particular kind of a disease and even particular kind of a target of for a disease, you can able to see how many mutations and what are the mutations generally happen in that disease. Because some diseases, the mutations play a significant role. So if you want to focus on the mutation informations, etc., I suggest you to go with this kind of open targets, which can give an extensive information on target of a particular disease. Okay, I just completed the session. So whatever I completed, just taking a theoretical part, I've taken this much time. Okay, so I show you. Can I take just for 5 minutes extra extra? So that I can show you something. And if whatever I didn't show you on today, I'll be exploring it on forthcoming sessions. Can I take 5 minutes extra on today? Yes or no? Only one.
Yes, sir. Yes, sir.
Yeah, okay. Any doubts at present? So this is actually my company website. So about company, I can explain you later. Now let me take you to the thing. Okay. NCBI, if you go to the NCBI, okay, National Center for Biotechnology Information, okay, this is the largest web resource present in the world for bioinformatics data. So if you see the big data present in the world, bioinformatics data ranks number one when compared to any other information technology data. And if you see in the bioinformatics, even whatever I call the father or mother of bioinformatics, she hails from this NIH. Actually, she is one of the director of National Center for Biotechnology Information. Here you can see plenty of information in this NCBI database. In this NCBI database, if majorly we work on protein targets, so first I show you from the proteins. If I type a protein called insulin, okay, in the protein, so generally it takes you to the GenPep. The GenPep database, I told you, whatever the GenPep information you have, okay. So we have different formats actually. The GenPep is a kind of a format. If you open there and whatever you see here is a kind of a GenBank accession number and what is the size of the protein this information you can see. This is a partially annotated data format. This is no annotation data format. That is FASTA. If I click the FASTA, this FASTA format can give you the first line. You can see the identifiers. The first identifier we see is the accession number. The next what we see is the locus name. In the locus name, we can see the protein name, as well as here you can able to see the species name. So the first line is going to have only the identifiers of the data. And in the second line, you can able to find the sequence. In this case, we search for protein, so we can see the amino acid sequence. The amino acid sequence, why we have multiple lines, Oh, I just mentioned you only one line we have. Second line alone is a sequence, but we have multiple lines. The reason behind is here it's a kind of a wrapping the text. You can have only 60 amino acids in the line. So because to see for a user friendly view, okay, so the database is developed in such a way that after each and every 60 amino acids, the wrapping of text is formed. So only you find two lines in the FASTA format. And this is what we call it as no annotation data format. And however I searched on a protein, similarly, you can type with the same keyword for a nucleotide. You can search for a nucleotide, you can search for a DNA, RNA for the particular protein. Okay, I told you even what are the types of DNA and RNA sequence we can able to get. So initially I got linear mRNA, then I'm getting linear DNA. If you scroll, you can also get the circular DNA somewhere. Okay, so these are some of the types of nucleotides, and if you want to focus on particular species, you can use this database filter. If your primary target is to focus on the Homo sapiens, so here around 11,754 sequences are available for insulin. Okay, so like this you can able to search in this gen pack. Similarly, if you want to search on a sequence or a I mean, 1D to 3D formats of a protein, chemical compound. You can search for PubChem compound. If you go over with the PubChem compound, you can type a name of a drug or a plant compound. So in this case, I typed aspirin, which is also called as acetyl salicylic acid, which is a pain reliever. Okay, you can get around 132 hits. I just typed a single compound, but what is the reason? So whatever I found, the derivatives of aspirin. The first thing is showing you the exact compound. The next are the derivatives. So you can find different kind of derivatives. Okay. So you can able to find this compound information from this pumpkin. So more about how to download 1D, 2D, 3D, we'll be seeing in the subsequent sessions. And we can see the genome database. This is what majorly we use. And for structure, we have to go with a database called structure, take you to the MMDB, molecular modeling database. So inside that, I just typed a PDB ID, whatever I know, 1XIH. So actually now we got some information about the 1XIH. This is something about the D-xylose isomerase. Okay, so most of the protein structures are complex with the ligands, some drug molecules. Here also in this case it is complex with the binding substrate and analogs. So about the protein structure, the online protein information, to download CN3D visualizer, you can use it. But you can, if you want to download the structure of that PDB, you can click this download. So it directly download the PDB file. So whatever we download, we can, I better create a kind of, uh, in desktop, I create a folder called AIMLDVD. So hereafter, I'll be using this folder for illustration. Okay, so the structure I have downloaded. Okay, so today I've just completed on how to download the sequence and structural information. Okay, and related to the protein-protein interaction, pathway databases, and target databases, disease information databases, we'll be seeing in the subsequent sessions. Anyhow, we have to use that in the subsequent sessions. Yes, definitely. So I'll be pasting my LinkedIn profile. Any other doubts you can ask me. Now I have completed my session. Any doubts you can ask me at present. I believe in the WhatsApp I have sent you the kind of a schedule, like how I'm going to take. Okay, so today I've taken biological databases. Tomorrow I'll be taking you on the target identification, modeling, and validation. So in that kind of a schedule, you can see my profile. Okay, so course instructor profile is available about the products information. You can browse through this company. Yeah, yeah. Can ask me. Someone has unmute. Can ask me.
So I didn't receive
Any doubt?
this WhatsApp group invite. How to get it?
Okay. May I know your good name, sir?
Ramesh G.
You can, you know my WhatsApp number. You better
Right.
send me the message so that I can invite. And one thing for all the participants, like if you have given one email ID, okay, definitely I might have it have reached you. If you have given two email IDs, the one what you have logged in is not I have provided a rights. Okay, so
Mhm.
if I didn't provide a right, I can give it to you. Okay, so the whatever you request through the WhatsApp. Okay. Yeah. So, and even while completing the MCQ and overall feedback on the final day, remember, use your registered email ID. Don't use multiple email ID so that it confuses me also. So check your kind of a feedback form you have filled. Use only the registered email ID to complete the feedback as well as MCQ on the final day. So on the final day, I won't restrict to complete the test on a single day. I give 3 days, minimum 3 days, 3 to 4 days. I give time. Like instead of completing MCQ on 12th, I can provide you around 15th or 16th, so you can able to, because if any of them have missed any kind of a videos, you can watch the recorded videos and you can complete even MCQ later. Okay, so this kind of a flexibility will be there for MCQ and overall feedback, just for self assessment, so perform well also in the MCQ. Okay, any other doubts, technical doubts? Okay. Uh, if no doubts, we can see on tomorrow. So tomorrow, as usual, we'll be recapping the sessions. So join on time. It will be helpful for you if you just recap and start with something new. Okay, so I thank all the participants who joined on time on today. Tomorrow also, I'm anticipating the same from your side. Have a great day to you all.